In 1972, Dr. Richard Nazik at the NIH developed hollow fiber cell culture in order to make a more in vivo-like way to culture cells. He created a cell culture system that closely approximates the mammalian circulatory system. Over the years, FiberCell Systems has refined and improved the materials and applications for hollow fiber cell culture. It is only recently that the true power of hollow fiber cell culture is being realized. Hollow fiber cell culture is the method of choice for the culture of large numbers of cells under in vivo-like conditions and collecting what they secrete. Hollow fiber cell culture is based upon the principle that each of the thousands of hollow fibers inside the cartridge are filters, like drinking straws, with a controlled pore size between 5,000 Daltons and 0.5 microns. The fibers are potted into a cartridge shell. Media that enters the end of the cartridge necessarily goes through the insides of the fiber, and cells are typically grown on the outside of the fibers. Hollow fiber cell culture is fundamentally different from flask culture in three ways. Unlike a T-flask, there is an extremely high surface area to volume ratio. Our medium-sized cartridges have 3,000 square centimeters of area in a volume of only 20 mils. This permits the culture of cells at very high densities, 1 times 10 to the 8 cells per mil or higher. These unique culture conditions permit the use of a simplified serum replacement, such as FiberCell's chemically defined CDM-HD, and results in significantly cleaner harvests, more controlled medium composition, and in many cases, reduced apoptosis. Unlike T-flasks, cells are bound to a porous support, not a non-porous surface. Cells are free to grow post-confluent, and splitting of the cells is not required. Culture and production can be carried out for many months in a continuous fashion with no change in productivity. The molecular weight cutoff of the hollow fiber can be 5 kD, 20 kD, or 0.1 microns. The molecular weight cutoff can be used to control the effects of secreted cytokines and also to concentrate desired secreted proteins, viruses, and antibodies produced by the cells. Concentrations of 100 times or higher can be achieved. Hollow fiber bioreactors require constant recirculation of medium to provide nutrition and oxygenation support. This is provided by the FiberCell Systems Duet pump. The FiberCell Systems Duet is a simple, compact pump system designed to support 1 or 2 of our small, medium, or large cartridges. The pump utilizes a unique positive pressure displacement mechanism that utilizes 2 one-way check valves to generate flow rate. The loop of silicone tubing wrapped around the back of the flow path stand provides passive gas exchange. Think of it as your heart and lungs. The Duet is designed to fit inside a standard CO2 incubator. The thin flat cord goes between the glass door without gas loss. Temperature control and gas control is provided by the incubator. Daily maintenance consists of harvesting the product using syringes and checking the glucose level in the reservoir bottle. The harvest volume is typically 20 to 40 mils. The glucose is monitored by taking a sample and measuring using a simple glucometer available from any drugstore. The rate of glucose consumption, expressed in grams per day, is a direct indicator of how many cells are in the cartridge and how well they are growing. The amount of glucose present in the medium indicates when it is time to change the medium for fresh medium. The medium-sized cartridge can support the same number of cells as 50 or more T75 flasks or 20 to 40 roller bottles. The larger sized cartridges can support as many cells as 100 to 200 roller bottles. Hollow fiber bioreactors from FiberCell Systems allows the culture of more cells in a smaller space, concentrating secreted products under unique cell culture conditions not possible with any other method. Although any cells can be grown in a hollow fiber cartridge, it is ideal for the collection of secreted products such as antibodies, recombinant proteins, conditioned medium, cell co-cultivation, and PK/PD dosage studies. Hybridomas secrete a factor called TGF-beta that is highly inhibitory to their growth. The molecular weight cutoff of the fiber allows TGF-beta to pass through the fiber while retaining the antibody in a small volume of the extracapillary space in a very concentrated form, typically 0.5 mg to 5 mg per mil. You can see in these gels of a non-purified straight harvest that the antibody is clean and free of contaminating proteins when using DMM with CDM-HD. The C2011 cartridge will produce between 10 and 50 milligrams of antibody every 2 days, while the C5011 will produce between 20 and 100 milligrams of antibody every 2 days. Hollow fiber cell culture permits the easy expression of 10 milligrams on up to gram quantities of recombinant proteins expressed in CHOs, 293, S2, and other cell types. The concentration of product is typically 100 micrograms per mil or higher. Improved protein folding can result from these cell culture conditions, as demonstrated by this gel filtration chromatogram of a hexaramized IgG produced in CHO cells. When produced in standard flask culture, 40% of the protein is expressed as an improperly folded monomer. When the exact same cells are loaded into the hollow fiber cartridge, 95% of the protein is now expressed as a properly folded hexamer. 500 milligrams of protein were produced in 90 days in a total volume of less than 5 liters of harvest. We see the same excellent performance when used with CHO cells. This is an example of CHO cells using DMEM with CDMHD. The expression levels in this case were very high, nearly a milligram per mil. The lack of apoptosis can be seen in this gel of the unpurified supernatant. It is a very clean harvest. Co-cultivation of 2 different cell types, or more, can be done in hollow fiber. Because the cells are bound to a porous support, the stromal layer does not need to be split, and the high cell density can facilitate cell-to-cell contact. The molecular weight cutoff of the fiber can concentrate secreted cytokines. In this case, a raw collagenase digest from a human placenta was loaded into the cartridge. The stromal layer established itself, and the cartridge supported these primary human cells for nearly 4 months of continuous culture, all the while generating a population of cells that could be harvested and plated out to demonstrate viability. The round bodies of cells were shown to be highly reactive with CD120, a mesenchymal stem cell marker. Another application for hollow fiber bioreactors is to retain bacteria or cells with virus in order to test different dosage profiles of antibiotics, antiviral agents, or anti-cancer drugs. The medium or broth recirculates, and drug can be added or diluted out of the central reservoir. Concentration of the drug can be controlled in a dynamic, time-course-dependent fashion, mimicking the precise kinetics of human bioavailability. The effectiveness of different dosage profiles can be assessed, dosage regimens that generate resistance can be determined, and 2 drug models can also be used. Hollow fiber cell culture from FiberCell Systems is the method of choice for the production of 100 milligrams on up to gram quantities of monoclonal antibodies and recombinant proteins. The harvested product is highly concentrated, up to 100 times higher, and is free from contaminating proteins from serum when used with our CDMHD. Protein folding is improved, and perfusion production can be utilized. CDMHD is a simplified, cost-effective, chemically defined serum replacement that is optimized for hollow fiber and other high-density culture methods. Hollow fiber bioreactors allow any lab to produce meaningful quantities of antibodies and recombinant proteins, as well as providing a unique environment for cell co-cultivation and other cell culture techniques. Hollow fiber cell culture allows any laboratory to culture more cells in a smaller space than possible using any other method. Available on our website is our video CD manual containing our video manual and a complete list of references along with other information. Visit www.fibercellsystems.com to request your copy today.